Evolution serves not only as a way to explain the great biodiversity we see in the world, but also how life has evolved around us through a collective series of changes to reach this point. A clear example of such change over time includes bird beak adaptation. As a population's food source changes, so can those animals' features evolve for new tasks. Here we can see a few examples of beaks best served in a transition from a plant-based diet to a carnivorous, insect-based diet, which would involve changes over many generations. In addition to large-scale changes like this, we can also zoom into the molecular level and observe similar patterns of evolution. Here, we can track the changes of molecules like DNA and proteins that have similarly evolved over time. Much like the biodiversity we see in the world across organisms like different animals and plants, we see similar variety on the molecular level. For example, even across seemingly very different organisms, there can still be proteins that are shared between them that perform similar functions and compositions. These groups of proteins are called protein families. With the advent of revolutionary sequencing technology, we now have access to millions of biological sequences. Starting from a single protein sequence, related sequence information can be found from its protein family. Combining all of these together, multiple sequence alignments, or MSAs, can be built to compare all the similarities and differences between these proteins and their amino acid composition. Using this data, information from thousands of different proteins gathered from many different organisms can all be used in analyzing how change has evolved over time on the molecular level. When evaluating these protein sequences, different statistical patterns of information can be found. Looking at this example portion of an MSA, we can see that across these sequences, the fourth position consistently shares a uniform pattern. They all have a proline at this site. This pattern of sequence information is called conservation. In addition to conservation of single sites within a protein, we can also look at larger patterns across the whole sequence. Here, with more information for each protein, we see a different motif. Instead of having only one amino acid in the same position, we instead see multiple amino acids at any given position. Yet if we look at two specific sites in tandem, a new pattern appears. While the single position might change, it always changes at the same time as the second, and we see the same pairs of amino acids showing up with each other. In this example, we always see a leucine showing up with a tyrosine, or a threonine showing up with a leucine in the second position. This pattern of sequence information is called pairwise couplings, as it considers how positions may have the same patterns of changes in pairs. Used in combination, this information can be used to perform direct coupling analysis, where both the conservation and site couplings are used to learn the relevant patterns of information from a given protein family. These statistics are used to calculate the Hamiltonian score of a given protein sequence, which informs how likely a given protein belongs to the greater family of related sequences. This information is used to calculate the global probability distribution and the statistical parameters of important sequence information for this specific protein family. Taken all together, we now have the relevant statistics of sequence information, as well as thousands of native protein sequences that have a shared function within this family. On its own, this information already gives us valuable insight into how these proteins evolved over time to have such diverse features across different organisms. But what if we could also utilize this information proactively? With this goal, we can turn to SEEC, Sequence Evolution with Epistatic Contributions, an evolutionary computational model for predicting protein sequence change. This evolutionary model begins with selecting a key family or protein of interest. Here we have TEM1, a popular antibiotic resistance enzyme found within E. coli. From the protein of interest, we can use the amino acid sequence to simulate an in silico evolution based on predicted sequence mutations. In SEEC, a position from the protein sequence is selected at random, and the relevant family statistics from the related sequences are used to inform what kind of changes could appear at this given site. Then, iteratively through n number of steps, potential mutations can be introduced across the whole protein depending on the simulation and parameters created from the original protein family. On the left, we see a simple example where after a given number of step iterations, a trajectory of the Hamiltonian score can be plotted for each variant created based on mutations introduced to the original protein. On the right, we see how each change introduced through the simulated evolution can result in various mutations accumulated across the entire protein. From our experimental results, our team has found that of variants we've created of TEM1, the evolution can accumulate 30, 40, even 50 plus mutations and still retain expected function as an antibiotic-resistant enzyme. This kind of evolutionary computational model is vital for not only further understanding the current landscape of protein information, but also where evolution may continue in the future. Considering the magnitude of sequence data available for use today, a vital goal remains on how best to utilize all this information. As well as the benefits of understanding the history of evolution within these protein families, data and models like this will further progress the fields of pharmaceutical drug design and treatments, immunology, and tracking viral evolution, and vital worldwide applications such as vaccine development. In utilizing the information we have at present, we step closer to creating the future we need.